In this video, we'll talk about blood sugar homeostasis, but particularly that part of homeostasis which allows sugar uptake in the tissues. Remember that as blood sugar increases, the body's goal is to decrease the concentration of sugar in the blood. There is a set of cells called the beta cells within the pancreas that produce insulin. Insulin tells the liver to stop adding sugar into the blood and tells most of the tissues in the body by volume to take up sugar from the blood. So you have two effects here. You have less sugar being added to the blood, more sugar being taken away, and that brings you back to homeostatic levels. Now, we talked in a separate video about how insulin is produced as a function of blood sugar. In this video, we're going to talk about how muscle cells take up sugar, or fat cells, as a function of insulin, or the lack of insulin. So, a muscle cell has a pretty continuous, very continuous uptake of sugar when insulin is not present. So it has a series of constitutive channels, so that as blood sugar travels from the blood, right, it's being delivered from the diet or from the liver, whatever, into the interstitial fluid, and then it's present in the interstitial fluid, it's got to get into the muscle cell. So the channels that are present when insulin is not present are called constitutive channels. And if you suddenly double the amount of blood sugar or triple the amount of blood sugar, because there are only a set amount of these channels, and these channels have a kinetics that are working at Vmax already, as you increase blood sugar, you do not increase the sugar uptake by a muscle or a fat cell. So let's look at what that means on a graph. If here is blood sugar, and blood sugar, of course, is going to dictate interstitial fluid sugar, then the rate of uptake by the constitutive channels has to reflect what we just said. So it must have a Vmax that is operating throughout all of the, that's supposed to be a straight line, throughout all of the range of blood sugar that you're likely to see within the body. So as blood sugar goes up, uptake into the muscle or fat cells will not increase. Now, that's not what happens in a healthy person, because in addition to that channel that we just talked about, here's the constitutive channel, it's still there. So when there's no insulin, you have this set amount of sugar coming into the muscle or fat cells. It is not dependent on the concentration of sugar outside. It's dependent on the Vmax of these constitutive channels. When insulin is released by the beta cells, it travels through the blood, diffuses through the interstitial fluid, and is bound to this receptor. So this is the insulin receptor. And the insulin receptor sets off, when bound to insulin, a signal transduction pathway that takes this vesicle full of another kind of channel. These are the insulin-induced, or GLUT4 channels for sugar uptake. Takes these channels and tells them become part of the membrane. How does it do that? It basically exocytoses part of the membrane. And because it's part of the membrane, all of those channels are now part of the membrane. And now tons and tons of sugar can get in. When insulin leaves, these channels endocytose and reform this vesicle. So if you were to take away the insulin, you would take away these channels from the membrane and they would become just part of this vesicle. So let's take a look at the kinetics of these channels that are insulin induced. They allow more sugar uptake, but they do so in a way that is sugar dependent. So the concentration of blood sugar does influence how much sugar these channels take up. Now, you're not going to get any of their activity at fasting blood sugar levels because there's no insulin around typically at fasting blood sugar levels. But, in a petri dish or something, if you treated those cells with this insulin, then you would indeed get that much sugar uptake through these channels. So they're present when insulin is present. They're not present when insulin isn't present at any blood sugar level.